we're gonna do a slight change of heading to 9 degrees, okay?
9 degrees?
Yeah, 9, 10. Yeah.
I don't remember reading about this in the plan, Albert.
Changing to 9 degrees.
Bridge nav, can we change the bearing to 9 degrees, please?
Seems like a big change.
9 degrees bearing,
We
please.
can just drive Hercules that distance.
Come on, guys. Everybody has its thing here.
Precision.
Must be precise. The only time we can deviate from the plan is to be more precise.
Bridge nav, can we step, please?
Can you step 19.76?
I have my arm folded most of the time, just to make it clear.
Just
Relax.
for the record, Albert has been a spectacular navigator.
It's 6 degrees.
Like,
you know
really
how,
amazing.
Do you know how many meters 6 degrees at 500 meters are?
How many? How many?
Right.
6 degrees at 500 meters. Just do the sinus and multiply things. I don't know.
Oh,
how
we
many.
only do trigonometry at night.
Just you know, take the sine, multiply it a few times.
Oh, he's getting the calculator out. He's going to show you.
Yeah, calculate it.
Kids at home, this is why you need to know your trig.
That's right. It's true.
Oh,
Otherwise
I thought
you
meant
can't show
over a
your
20-meter
friends.
step, not a 500 meter.
That's 140 meters.
Nobody's
way.
got anything to say.
Yeah.
Yeah. So
Oh, I thought you were doing 10 degrees and then you went to 9. You were at 15 or something?
it
No,
was
that-
15
I was
to 9.
15.
Yeah.
15 to 9, oh,
Albert,
So there.
I concur with your calculation. I was just doing it in my head at the same time.
Yeah.
I figured it out independently.
But I got about 140.4.
Yeah, I figured just rounding errors.
His floating point error. Sam doesn't have that.
It's bad. It was 52 degrees though. 52 meters. I had my angles in degrees, in radians, not in degrees.
Ah.
Oh,
Still 50 meters, you know? It's like 10%.
But who knows what we could see in those 50 meters.
Is it within the error of our contour drawing algorithm of the multi-beam data?
Bridge nav, can we step, please?
Should have the viewers do a calculation for us.
Have the viewers do it.
Yeah, next time we have a dispute to
We'll-
settle, we'll have the folks at home do it
Yeah.
for us.
A neutral party.
Folks, do the math. You give us a heading.
Albert's always doing things in metric. It gets confusing.
And then have them calculate at this ship's speed what the time difference would be to get there
Oh.
versus
If the
Ooh.
train left Chicago at the same time the Nautilus
right.
left Samoa.
Oh, no.
when you were
You
looking
know,
at it from a hot air balloon 10 miles above the surface.
Which is actually like relativistically like
Yeah.
Factor in gravitational lensing. Everything's fine.
Oh man.
It's 88 miles per hour at 1.21 gigawatts.
Are we taking air resistance into account?
Oh my gosh.
Can we zoom on this yellow guy, please?
yeah, go ahead, video.
Thanks.
back to the science that we
Yes,
actually
we're
understand.
Diving on unexplored sea.
Is that a paragorgia?
Which? The yellow?
Yeah.
The yellow I think is a plexard. It could be a Canthogorgia, but
Okay, come
it
on.
does look a little Canthogorgia-esque.
How would you spell that?
Oh, I would never.
I'm spelling it.
Let me see. your-
Accurately, I'm not sure. That's my actual job.
hold of your next move, Albert. I'm just
That was
struggling
good. That
to-
whole evolution of whatever nonsense took 6 minutes.
Just flying by, really.
It has been 6 minutes since I last looked at the clock and wished there was a bathroom up here.
Yeah.
Oh, no.
You see, do
We're
you want to
getting
change 1
there.
degree the
We're
heading now?
close.
That gets you
And-
6 more minutes.
And argue about it a bit too.
We could put the same bathroom they have in Elvin up here.
You mean the bucket?
We
Yeah, it's
all
a-
have to agree upon that one.
It's a very nice bottle.
Oh, God.
We don't talk about those things.
I have a question for our scientists from a college student who's watching, and he is a biology major and is wondering how you guys got yourselves as scientists onto research on research ships and any advice you have for him to do the same.
Well, for me, I started going out to sea in graduate school, so I did my PhD after undergrad and was working in a deep sea lab with Eric Cortez at Temple University, and essentially, since my advisor had funding to go out to sea, I was able to get on a few cruises. The Nautilus also has an internship program that I'm sure Kelly could talk more about than I could.
Mm-hmm.
Should be another way.
Yeah, that's a really great way to get aboard the Nautilus, especially if you don't have any experience. You don't necessarily have to have been at sea to come out here. And if you're on the Nautilus Live webpage, right above where you can watch the streaming, we actually have a link to our application. So those are due every year, usually around December or January, and they're open now. So you should look at them because we do have undergrads who come aboard. We have an undergrad science intern right now, Gina, who's on a different watch.
Good for step now?
But yeah, that's a great thing to watch out for and a good opportunity to
Or
get
you
aboard.
wanna wait some more?
It's pretty steep
Just a
here all
second.
of a sudden.
Yeah, and I'm struggling to make any ground.
Okay.
here. Hold
Yeah, it's
on.
funny. I got an email when we were out, like this week, being like, "You could be the next person to sail on the Nautilus" or something.
Did I get this in?
I'm here.
You got that
We're
mail?
already here. We have to jump on that.
I made it.
No, I would say if the student's interested in oceanography, they should go to graduate school at a
Yeah.
school that offers an oceanography program, and then you'll definitely, most likely, get invited out on a research cruise.
Yeah, when I was looking at graduate programs, I was specifically looking for ones that would have field experiences and cruise opportunities.
Can we stop and do a closer zoom on any one of these white corals up here?
Yeah, I'll get you something. That'll
And
probably
then
be
you
pretty
ended up with
shaky
mud.
here.
Okay. I mean, I think we're gonna keep seeing them too, but
Okay, zoom in video.
I've been on a bunch of cruises with undergraduates as well.
Yeah.
And so like if you can find a researcher at your university who is interested in like seagoing types
yeah,
of
this
research,
looks like what we sampled earlier.
you
Thanks, Mike.
sometimes can get out to sea
Okay,
with them.
Full wide.
Yeah,
please.
so like in my university at Oregon State University, since it's an oceanography college, for the undergrads in the program, we have each year a week-long course where we take people out on our research vessel, undergraduates out on our research vessel, and they get to experiment with lots of different oceanographic equipment and things like that. And a lot of oceanography schools have that. But if your school itself doesn't have that, then internship
That's a lot
programs
of coral
are really
gravel.
great.
Yeah.
It's also great to learn early on how seasick you get.
Yeah.
I knew a grad student who dropped out. Majority reason was because she just got insanely seasick and just couldn't do field work.
Oh, that's so sad.
But for most people, it's not too much of an issue.
Okay, Albert, you can
Step
call that
now?
move, yeah.
My wife gets seasick just looking
Reach
at
now.
boats.
Really?
Yeah, my wife too. She would never, ever come
Gabby,
you.
do you want to speculate?
We did
on
this
what
dolphin
kind of,
dolphin tour
snack
last
they
year
made
in
for us?
Hawaii or a few years ago, and one of the staff came up and he was like, "Do you know your wife is like in the back looking really sick?" And I was like, "Oh no, I know. Like I'm up here looking at dolphins." Like I've very clearly made my choice, sir.
Yeah, I'm aware. I'm aware. Just
Well, I
That's
couldn't
funny.
cure her. I'm not that kind of doctor. I actually think it's very frowned upon for me to practice medicine.
That's funny.
Well, I'm still waiting for someone like on a plane to be like, "Is anyone here a doctor?" And I'm like, "Yes."
Oh wait, you want me to do what? No.
Yes, oh, no, not that kind of doctor.
Oh, sorry. Is this
Sorry.
not a deep sea ecology thing?
Okay, zoom in there, please.
I am an engineer, but I don't want to be on a plane when somebody says, "Is anyone here an engineer?"
All right, forward.
I agree with you, Gabby.
I don't think anybody wants to be on a plane when that.
Anyone here an engineer?
Yeah. Is
That'd be
anyone
a great
here-
prank from a pilot.
Does
Though.
anybody know anything about fixing planes?
Is anyone here a pilot? Well, I'm an ROV pilot. Does that count?
Yeah.
Step.
Yeah. Yep.
Bridge nav. Step again, please.
Yeah, I'm not that kind of doctor, and I'm not that kind of pilot either.
No, you just quick. There's just disappointment mounts by the second.
I'm sure that some skills would transfer.
Yeah. I mean, if you had to.
I had somebody introduce me to their friend's their 3-year-old son, maybe 3, something 2.5 or 3, and just saying that because he was really into watching some underwater science show on TV or something. And she's like, "This is Michael. He's a submarine pilot." And then I'm like, "Well, no, actually." And then he's like, and then he starts, right? He's just like, showing a few words like, "You go in the water," and all this. I'm just like, no, it's a robot. And then it's like, "Oh, is it hard to breathe?" And it's like, no, I'm on the ship. It's a robot. And then as he understands it, he just like starts looking in the dirt. You know?
That's so forlorn.
That's stupid. Real job.
Get a real job.
Goes from amazement to just scorn.
There's no scorn like a 3-year-old scorn.
Yeah, it's like, this guy's still playing video games. Idiot.
What's the show they watch?
Aquanauts. Yeah. Aquanauts.
Oh my gosh.
Another move.
We didn't do
still
that
doing
move.
it.
Yeah, we didn't do that move yet.
Yeah, we're all okay here now, I think.
Yeah, I'm pretty happy here.
Albert, what did you calculate for the ascent? An hour and 20 minutes or an hour and 50 minutes? I can't remember.
I don't remember.
Like about 80
80
minutes.
80 minutes.
Yeah.
80 minutes.
I think we just rounded
All
to an hour and a
forward
half
call.
with like vehicle positioning and stuff. And we have to fire that analogy on the way up.
Stretched out.
Roger.
Move in the works, though.
Bridge nav. Step again, please. 1,600 meters we've done
Just
Oh.
on this watch?
Wow.
just on this watch. Yeah.
Wow.
We've been really disciplined.
Yeah, we've
Yeah.
only sampled a few things, and we've been moving.
Very good, very good.
And Albert's been cracking
watch,
the
good
whip.
Yeah, sticking to the plan.
We haven't been playing with the crowbar.
Yeah,
Oh,
I know. Sam, I'm with you. I think this is a colossal disappointment. Just devastating.
that's the only word for it.
All right, team. We have to make it a mission before the end of the last watch to play with the crowbar.
To use the crowbar?
We got to find a reason.
Maybe it.
The problem is it really comes down to Dwight, because Dwight's the rock guy.
I
Like
know.
Sam wishes he was.
If we could use it for corals, we'd use it every time.
Yeah.
You guys would be like, "Oh, I think we should slurp," and I'd be like, "No."
No, we're
definitely
gonna need
more
to
of a
use the
crowbar.
crowbar.
Bridge snap, step again, please.
I think we've been pretty effective without
Do you think we can move back to 40 meter step?
We can
That
try.
shouldn't be that
Yeah,
sloppy
we should
anymore.
try
try
it
it.
now, yeah.
We will have 3 more dives, so
So next
Yeah,
step
maybe
will
at
be 40.
Johnston.
we don't have to do it in the next 10 minutes.
I just don't want to be on a plane going home and be like, you know, we never used that crowbar.
No regrets.
Oh,
you
that
know?
does sound really sad.
Flight attendant would be like, "Why are you telling me that?"
Zoom in, please.
You get home, you go to the store, you buy a crowbar.
It won't be the same.
It won't be the same. Eternally unsatisfied.
Yeah.
You start trying to articulate your arm like a robot.
Go in there more.
That's as far as you can go.
It looked like there was something in there.
Okay, come on.
A little sponge prisoner.
Wow, still 300
We would have
meters.
gotten to the end if it wasn't for that current.
Yeah, if
It
we
would have
didn't
been
stop
like a
for that
photo
sample.
finish.
Yeah.
Yeah, that's that one sample that we had to backslide and everything.
Yeah. Stop, go back, go.
And then I knocked the coral off and everything. That turned out to be a disaster.
Yeah. Bridge snap. Can we step 40 meters, please? 40 meters.
Yeah, I know. It's just the optics are terrible. I know. We used to work with, their silence was deafening.
All right, we're going to do a watch change.
Watch change.
Just, I think the shallowest location, you know, just to say we took one off the top. So it can be anything. Yeah, you could try to get a rock that has another sample on it.
Yeah. I mean, if you find like a new phylum or something.
big cat.
Hey everyone, we just got a quick shift change going on, and then Team Violette will be back with you.
Check, 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 check.
Ben, you've got the lowdown that we're just
Yes.
going.
We're just going. 40-meter steps. I'm pretty happy with that.
Trying to just get to the top of that. Yeah, just like
Feature.
the end.
And so after this, we'll go to the summit, which is also about maybe 200, 300 meters off. So
If we have time.
okay, cool.
But if we don't, then we'll just stop
Explore
it.
and
Yeah.
Great. So close.
So close. Yeah, we got some corals. And some rocky rocks.
Unlike
As
non-rocky
opposed to,
rocks,
yeah.
you know, like the
Sandy
sandy
rocks.
rocks. Yeah.
These are really like angular, not so.
Not very grape-like. though.
Encrusted. They're still encrusted.
What were you gonna say? Not so what?
Remember the words.
Grape.
But
Not so grapey.
Pillow?
pillow.
Oh, botryoidal.
Botryoidal. Not
Botryoidal.
Not like grapes.
Botrimodal.
Close. There's no mode. Remember the Greek.
Botryoidal. Okay, okay.
There's a Y in there, if that helps.
That does help. That does help.
Botry, the Y.
Botryoidal.
And an oidal.
Trimodal.
I need to learn how to spell that.
Botryoidal. Moidal.
No, don't confuse me. I just got it.
Bacchus. Bacchus was the god of wine. Does that help?
Oh. Are we waiting on the ship? Sorry.
Not quite.
Okay.
What'd you say?
Hopefully we won't do a curly Q.
What was? Botryoidal.
Ben, what was your bearing again? Sorry, I was doing
I'm going
numbers.
due
Oh,
north.
there's some rubble.
Due north, roger.
Yeah, it's a lot of rubble.
Little rubble patch.
Trouble.
More rubble.
More rubble. Look at that.
I see rubble in bubble.
Rubble.
hope not,
Bubble's
'cause
been
we're gonna
stolen.
get
No, bubble's been stolen. Background rubble in
It's
bubble.
number time.
Background number and bubble. There you go.
It's becoming like spoken word.
With some sprinkled in Greek and Latin.
Ooh, let's take a look at that bigger white one up a little ahead of us.
So do we,
We
have
can't
room
hear
for
your
samples?
enthusiasm. We got one water and one rock.
Ooh.
Yeah, we're not sampling, but we did sample this white one down below.
We're just exploring.
But I'd like to take a zoom on these. What's our depth?
1170.
On vehicle. There we go.
1170. Interesting.
All right, video. Wake up and zoom.
Yeah, I'm here, Spicy at the beginning of the dive ship. Ooh.
There's 3 different species?
Yeah, looks like it.
What's going on the leftmost one there?
So the leftmost one and the other white one are likely the same, but the left one has all of its polyps out, and the other white one has all of its polyps retracted. It's interesting because we sampled one earlier, and the skeleton was a lot harder than I was expecting, because based on the texture of the tissue, it looks like maybe they would be within the family Paragorgidae, which normally has like a spongy skeleton and is bendy, but it was kind of brittle, so it broke. So I'm interested to see what it looks like when we have it on the surface.
Greg, I'm going to zoom a little
And
more.
then the yellow on the
Sure.
left is a plexaurid octocoral. And then, yeah, that's great.
There's 3 different species.
Yeah, it looks like it.
What's going on the leftmost one there? So the leftmost one and the other white one are likely the same, but the left one has all of its polyps out, and the other white one has all of its polyps retracted. It's interesting because we sampled one earlier, and the skeleton was a lot harder than I was expecting, because based on the texture of the tissue, it looks like maybe they would be within the family Paragorgidae, which normally has like a spongy skeleton and is bendy, but it was kind of brittle. So I'm interested to see what it looks like when we have it on the surface. Sure.
can really see. Doesn't it look like it has kind of like gummy texture?
Tiny little
The other one?
there too.
No, the white one.
Just because of the color, I thought it was exposed skeleton.
Yeah. that
Oh
doesn't
yeah,
look
it's
gummy
not.
to
There's
me.
tissue on there.
It looks brittle. Brittle bones.
And then that's an Acanthogorgia, I believe.
That's a really cool color.
Yeah, this is a cool image too, with like the very fluffy polyps extended on the Acanthogorgia and the polyps retracted on that other coral.
All right, let's come full wide. Push back out.
Hey, Ben, you might want to reset the DVL again.
Yeah. Are you off auto? Yeah. Okay.
So we have about, and Greg, correct me if I'm wrong, about an hour left on the bottom.
Just do run it at 15 meters a minute, 1,000 meters.
15 meters. Well, we might be a little heavy.
That's
So
why
I guess we'll drop a weight.
Yeah, we're definitely going to drop a weight. Usually do 20, which is about an hour, but we're going to be
a little bit.
6:30. We should probably be off bottom in half an hour.
Oh, okay.
If not before then.
Cool. So we can't sample, so let's probably just cruise. We'll zoom every once in a while if there's something really different looking, but I think it's important for us to try and get at least to the top of this little hump.
We can't sample because we're at.
We
we're
have a full,
or-
yeah, we already sampled.
We are full up.
A lot on this
Loaded.
dive.
Yeah, I'm bringing us to 010.
Roger.
So we'll look for a rock when we get to the top. And probably just stash that on the porch on our way up.
Look like a Calicophron typhonophore in the
Yeah,
August.
the porch is the front of the ROV, if anyone's unaware.
Yep.
That's the porch.
That's the porch.
And the scoop.
And the scoop's hanging out on the porch. And we...
Well, where do you put your scoop?
Next to the kitty litter.
I leave mine. For all the deep sea cats out there.
I was going to say in my coffee. I feel like the closest thing I have with a scoop in my house is a coffee scoop.
Current's still cruising through here.
Yeah.
Ooh. What we got floating by there? Yeah.
Looks like a sea cucumber.
Flying sea cucumber. If you ever do much snorkeling in the nearshore environment and your experience with sea cucumbers are, you know, little lumps on the on the surface that you don't see move too much.
Or on the sediment, probably.
And this, yeah, hanging out in the, oh, not surface, sediment, yeah. Hanging out in the sediment. It's kind of cool that they will actually take flight.
Yeah,
And swim.
some of them.
Swim through the water column.
It is cool.
Do the species that will swim around and filter feed, do they look fairly similar to the stationary ones when they're actually on?
So there is one
sediment?
type that looks pretty similar to the stationary ones. Those ones we've seen, the Plagithoria, do not look similar. They don't.
They don't like fold back in to be.
No, but there is one that swims that does look like the ones that are just kind of lumps on the seafloor,
Yeah.
but it'll like launch itself up and then like undulate back and forth.
Oh, wow. That's pretty cool.
Might do it 020 because we want to get Argus kind of centered on that top.
Roger.
Might be at the end of your leash there.
Fine. We'll zoom on something.
Better.
With like 10 meters left in this move. I'll do 0.
Can we zoom on the rubble in front of us?
Sure.
Well, that is quite a lot of rubble.
Yeah.
Do you think it was consolidated here from being deposited here? Do you think there was a community that was at this site?
Yeah, I think that there was a community at this site. It's either that it's come from higher up,
Video
so we'll see
pushing.
if we see any evidence of that, or that it was just growing around these rocks. It's pretty old. We can.
Very dead.
It's very dead. Gets that dark coating from the manganese, ferromanganese that will. Yeah, so similar to rocks, it happens to coral skeleton too.
Interesting.
Does the rubble play into the ecosystem at all?
Yeah, it's hard to see here, but there's generally a lot of smaller invertebrates that will live within the rubble, and on other coral mounds that have been more extensively studied, it's been shown that there's actually higher diversity within the rubble than on living corals.
There's a little pincher
Yeah,
in there
yeah.
on the inside.
Right
So
on the
there's
top.
somebody living in there.
And there's definitely some brittle stars in there. We find all kinds of different small invertebrates that live within the rubble. That's interesting, rock.
I don't know if that's rock.
Oh, is that coral?
I think it might be sponge.
Oh.
Wow.
All right, video, let's come wide. I had never removed my hands from the joysticks, but I had hit stick lock without remembering. So I started driving again.
You're like, why am I not going anywhere?
This is pretty steep.
Yeah, it is. So are we just coming up? We're pretty close to the top of the seamount right now, aren't we?
Yeah, so there's like this little local maximum outcrop right below the summit that we're climbing on. So we'll go down about 30 meters off this and then come back up to the summit if we have time.
Okay.
Yeah, the summit is about
Giving you a bit of a tug here.
maybe 40 meters above us.
Part of that's current.
It's interesting. We're seeing a lot of these. I think it's a type of
I'm looking
corallium,
050
the white
currently.
corals. But we haven't really
About 30
seen
degrees off of our heading.
many, if any, scleractinian of the hard corals like what we saw on Howland Island yesterday.
Yeah, at the top. Of
there's a lot of bigger communities around 1,000 meters
You want me to stop the ship moving to
of
a
those
0.0?
corals. So quite interesting to see the difference in community between these two and Baker as well.
When we were on Howland at 1,000 meters, though, were we out of the basalt and into sort of the calcium conglomerate yet, or was that still basalt?
That was still basalt. I think the calcium started maybe around 600.
Okay.
I don't think we did intros. You ready, team?
My name's Greg, and I am here. All in attendance say nay.
Ooh, although, hang on, what's
It's a
that?
very large
Is that just a
crinoid.
crinoid?
Yeah. Not just a crinoid.
No, I mean, it's not just a crinoid. Like, it is amazingly a crinoid. Because they are super cool, like giant fan.
How
filter
spoiled
feeders.
we are now.
I know, we're so spoiled.
When I rank I have to assign like a numerical value to the photos, and I'm like very nervous that I'm going to start getting dull about it. Like, oh, another crying RV1.
Oh.
Another Dumbo octopus.
Yeah.
Ew.
Well, the nice thing is we'll have a couple day transit in between.
Yeah,
We'll
so we
reset
can
your
get bored
memory.
again.
Yeah.
Yeah, reset your memory.
Yeah. Then all this will be new and exciting again.
Just some of us, it takes longer than a few days. But
It's always exciting, you guys.
It is.
video, let's push in.
If I need to remind everyone, we're currently diving in remote operated vehicles 1,100 feet or 1,100 meters
Even
below,
more exciting.
below the surface of the ocean on an unmapped ancient volcano and beaming it live to homes around the world in real time.
And
This is
it's
why we
just
love
not
Holly.
enough.
It's just saying. It's just not enough.
Gratitude check.
Yeah,
And
sometimes
we're looking at hermit crabs that are on a glass sponge.
Dead glass sponge with the
in the
crinoid.
ocean. It's
Said
pretty.
dead
It's pretty
glass.
stinking cool, guys.
it is.
That's why we have an enthusiast.
Exactly.
Make up for the most
All right,
emo
video, let's
team
go
ever.
one.
That's the ultimate enthusiast moment.
That was really necessary.
I got pushed in the back here.
Every watch needs an enthusiast.
That's true.
It is true. Sometimes people ask, like, are you guys having fun? Or there's people hear us laughing and, like, there's some comments about the laughter. But it's like, this is, I mean, we're doing a dream job right now.
Yeah.
And we know it and we love it. So what's really great is that we get to come out with a really great team. None of us knew each other before coming on board. And we've been able to form this rapport where we get to do this exciting work together.
Front row and data bond, right?
Yes.
Yeah.
Okay, fine.
I was
Sorry.
gonna say just
We know
front
each
row.
other.
That's very
like,
true.
I can't
Sorry.
leave Megan out of that.
Fine,
That's
fine.
We've been here for a minute.
Yeah.
I didn't
Right,
know
time
any of
travel, I
you
forget
before
time
coming
travel.
here.
Yes.
And now we're the dream team.
As a team, we're
Video,
new.
push in.
Yes.
What's that little guy?
Tina Ford.
What is?
Oh, Tina 4, yeah.
Oh,
Hi, Tina 4.
Oh.
yeah, another Sedippetina 4. It looks like it might be a mertensid species.
Come
Its
here.
tentacles
Super
are retracted.
small.
Yeah, they tend to be small.
Oh, I like it.
Yeah, these have really fine tentacles, so even if they were out, I'm not sure we'd be able to see it.
Oh.
So, and those tentacles are.
there
Oh,
they
you
are.
can see one, yeah.
Oh, yeah.
Yeah.
Oh. So cute.
Ben, those tentacles are for filter feeding? Is that what is it?
So they're for
Style.
actually predation.
Oh.
Yeah. So
Really?
the teens, those comb rows, are kind of an either-or function for some species.
Stay
So
still.
they're kind of a form of locomotion. They have the teens are actually cilia that beat against the water and propel it. But other non-predatory species will filter feed using those teens and bring the particulates that are
All right, full one.
entrained in that
very hard to
into
focus
their
on.
mouth.
Yeah.
Yeah, it's small.
Yeah, it's super small.
Yeah.
It's very
That was-
far away.
That
That
was
was
a nice
well
job.
done, you
Yeah.
all. That
Yeah,
was
great
really well
job.
done.
It's hard to follow those animals that are swimming.
Yeah, you know what? This is too hard. Let's
Fix
just
to the ground.
I'm done.
Stick to the corals, everybody.
Stick to the core.
Stationary.
Says the coral biologist.
Yeah, right.
What are those swimming things? Where's my coral?
No rocks, just coral.
It's a pretty good interpretation or imitation of me, actually.
Real old timey.
It
I know.
was a little old timey there.
She's been around for a
Pretty
while.
old-timey. As we've established,
Yeah,
Alexis
I'm
has
trying
been
to
studying
follow
coral
the
for at least
way
3
up,
millennia.
You know I'm at 030 now?
Yep.
Okay.
Thought I was alive for 3 millennia.
Studying coral. For 3
Yeah, you were just a baby for 1 millennium.
Yeah. Oh, hello.
Wow.
And we still have not introduced ourselves.
Oh, right.
Except
I
for
introduced
Greg
myself.
did.
Greg did.
Greg's
Yeah.
on
Here.
it.
Good job,
He's
Greg.
present.
In attendance.
In addition to Greg being present, my name is Brandon Rodriguez. I'm the Science Communication Fellow, very present.
What does the Science Communication Fellow do, Brandon?
Other than color commentary and cracking sarcastic jokes,
Which you do well.
oh, thank you. I get to watch chat, see what you guys are contributing, and hopefully enjoying the feed here on the dive. I also get to interact with classrooms across the the U.S. so far, although I imagine we could extend beyond that. So if you are a classroom teacher or anything related to education and looking to have someone speak to your students, we're able to do that via the telepresence that you're able to watch this live feed.
Super cool.
And I mismathed. We do have about an hour.
Oh, okay.
So just to get to the summit of this, I might do a
Anyone else
10
present?
meter move 090 for this.
Video.
I guess yeah, I'm
Did you
here.
mismatch along with me?
I'm Marley
Wake up.
Parker. I am so
thought it was
stoked to
5
be
that we
here.
started.
I'm just a little bit tired. we've been. This is our third dive, or this is our third watch of this dive, I believe, if my
Oh yeah, that's
Yep.
true.
if I'm mathing correctly.
You are.
Oh, indeed.
Yeah, so my job's really awesome. I get to control the cameras on this thing. And also responsible for
All
the
right, we're going to move you to
26
east.
other cameras
See
we
if
have
can
in our
get the-
video
I
system.
just want to see if we can get them on the summit.
And every time...
26? I thought it was 27.
090.
Yeah, but I just was including the one that I'm controlling right now.
Oh, okay.
It is
The other
27 altogether.
26. There we go.
Science, do you want to know something funny? Not funny,
Yeah, because,
but-
well, I'm looking.
Okay.
Yeah, because
Yeah.
you're heading.
Those are the two
Why not?
tops, right?
It's really not that funny.
And I'm-
But every
I'm at-
time
yeah.
Yeah,
the ROV
065,
pilots
so.
check the numbers, I also do a systems check.
Ah.
You're one of them?
So
already there.
Whoa.
You do the
Extra
numbers?
numbers.
my numbers aren't as exciting, but they are important to make sure that everything is still recording and that our files are intact.
Cool.
Wow. Because how lame would it be if we did all this awesome footage
back
and
up a
the
little bit
files
while
weren't
that swing
actually
comes through.
there? That would be bad. if
Bad.
Something
That
didn't
is tough.
actually get saved or recorded.
Right.
Yeah.
We do a
So-
numbers check of the time, mostly,
Yeah. Time is important.
because it's always at the top of the hour.
Right, that's a lot of rubble.
Mm.
That is a lot of rubble. Man. Just and old.
Yeah, I think the summit's translated just a little to the west. So.
Video, let's push in.
Got a little Paragorgia right there growing in the rubble.
Glass sponge. Growing too.
Oh, hi, Paragorgia.
And there's purple in there.
Yeah.
There is purple in there. Let's see.
Is that the fluffy purple or the Victor Gorgia?
Might actually be what's called.
That
It's
encrusting.
either, yeah. Well, there's also a polyp right there, so it might be a stoloniferan, which is like a crawling coral, so they don't form their own skeletons,
Yeah.
but they just grow across substrate. Oh, there's a little,
Yeah.
little glass sponge.
There's a little bivalve.
Yeah.
2. 3 little bivalves.
Yeah.
Sitting in a row.
Trying to look at the branching of the
Probably, yeah, because we want to go up and over the top.
rubble. Looking
Yeah.
to
So
see if I can
we didn't
figure
want
out
to
what
rock
kind
here.
of coral
You
that's
wanted to rock
from.
up here? Is that the idea?
Well,
It's a lot of it,
not
yeah.
at end. We want to get a rock. Megan, do we want to get a rock at this peak, or do we want to hope we get to the next peak and then get a rock?
I was kind of hoping for the next one, but if
Okay.
we see something nice.
All right, I'll keep moving.
So then, yeah,
forward
let's
then.
keep moving.
Yeah.
So after this dive, we have a couple day transit, so we won't be diving. We'll be heading up to Johnston Atoll,
So I'm facing 090 currently.
which will take us about 3 days to
Yeah,
get there.
he's
So
moving you a
we'll
step over.
be updating the Nautilus Live status, but just keep in mind we won't be in the water for a couple days.
But we will be mapping,
We will definitely be mapping.
and you can watch our mapping live.
Ooh,
Which is pretty
You can.
exciting. It's surprisingly exciting.
Rainbow Road.
Rainbow Road.
Row.
We'll also work to put together some of the best photos so far in the first half of the trip, so keep an eye on the
Oh, cool.
albums.
Yeah. Do we make it across the front row with the
Oh,
intro?
sorry, no. My name's Jess. I'm sitting Argus right now. What was the other thing we were adding in?
We were kind of
Oh,
talking
I'm
about what we
currently
actually
present.
do.
Oh, yes, yes. Yeah, part of the ROV team, so we maintain the vehicles, and then when they're in the water, we fly them. Yeah, so that requires a bunch of wrenching sometimes, and when you're in the van, it requires different types of concentration. So it's pretty fun though, either way. All right, Greg, round 2 for you.
Round 2.
Oh.
My name is Greg, as introduced by Jess. And I do pretty much what Jess already said.
Sitting Hercules.
Yeah, I'm Ben. I'm sitting in the navigator seat.
Video, let's push in.
Is that
It was a purple crinoid down there.
goth crinoid?
Yes.
Goth crinoid.
Just misunderstood crinoid.
There's been a couple of them, but they're sitting on a rock. They're not up in the water column at all. All right, full line. And let's push out and get lined back up for north.
There's so much coral rubble.
I know, and I haven't seen any live coral.
Yeah, it's pretty weird.
I mean,
It is
there are little
weird.
live corals,
Graveyard.
but there's no...
Yeah, but they're not the same type as what the rubble is.
Oh, really?
Yeah.
Oh,
And the rubble's pretty old.
And the rubble's pretty old. Interesting.
Yeah.
It's a mystery.
My intuition would be that there was something like external to the coral that kind of made them die, like something not in themselves. But how often do you see or do you know of any kind of diseases that hit a single species of coral? Like the most I've heard of this kind of thing was like sea star wasting, and
Right.
things like that.
So in deep water, I don't really know of any
Yeah,
I'm gonna
diseases.
start heading down some too.
There are shallow water coral diseases.
Right.
I can't remember what one is called. It's like white something. It's not bleaching, but it's like there's a
white something.
There's like you get these white patches on some gorgonians in shallow water.
Okay.
But I don't know of anything like that in the deep sea. But again, we also just kind of have glimpses, so we're not watching in one spot long term in a lot of places. There's a couple places in the world where people go back pretty regularly, but environmental conditions are really important for them. So temperature is a huge factor. They have a pretty narrow temperature range of just a couple degrees Celsius, and then like the water acidity is also acidity is also really important. So it could have been that these were growing a long time ago, and then conditions changed, and so they died off, and now they just remain as rubble. One of the projects we have going on in our lab is looking at how quickly that rubble dissolves. So we have a dissolution experiment running just to try and understand like what's the rate of dissolution under these conditions, to try and extrapolate some information about how long ago they may have died. Hold on.
Wow. And do we know much about like how long it takes that ferromanganese
what's
crust to
that?
form? Ooh.
Like a radiolarian?
What are you? Pincushion.
There it is.
Yeah.
Hi.
Might be a radiolarian, but it's really large.
Yeah, I was gonna say.
What is it, Ben?
A radiolarian, they're like single-celled protists, but they can be quite large. But they do have those like kind of projections. But I'm not sure what that is.
Well, too much.
Sorry, y'all.
There we go.
Oh.
Oh no,
Huh.
Interesting.
There are weird things in
What
the
you
water.
know, yeah.
A really zoomed up look at pollen, but it's not obviously. But
But that's what radiolarians kind of look like.
really,
Yeah.
Yeah, or popcorn.
Yeah.
Alexis, to the ocean acidity question, the idea isn't that the acidity is directly, harming the coral the way it would like us, but that it changes growth rates, right? We're not talking about orders of magnitude change. We're talking about kind of ion concentration change. Right?
Yeah, definitely. And you have a chemistry background, so you might even be able to explain this better than I can. But it's just that when there's more CO2 in the atmosphere, the ocean is taking up more CO2, and it just makes more hydrogen ions within the water, which decreases the acidity of the ocean. Is that right? Increases the acidity, decreases the pH.
Right.
There we go.
Yeah.
And corals are calcifying organisms, so pH is really an important factor for them.
Video pushing.
And
Is
it's
it Brazilian?
the zooxanthellae that gives it color that they expel.
For bleaching?
For shallow water.
For
shallow water?
Yeah.
Yeah. Yeah, and most bleaching is induced by warming.
Right.
But yeah, so like shallow water corals have little algae that live within them that I recently was told we should be calling them Symbiodinium and not zooxanthellae.
Okay, cool.
I know, Sarah told me that
I think there's Symbiodinium.
yesterday.
Zooxanthellae.
What?
Symbiodinium, and then I thought the species name was
Zooxanthellae.
No,
Wait,
Symbiodinium, zooxanthellae like we used to be
Right.
an actual taxonomic term, and apparently now it doesn't hold taxonomic weight anymore. But Symbiodinium is the group of all of those algae that live.
Oh, okay.
Yeah.
All those high school textbooks.
I
will
know.
be changed.
I know. Well, it's also like a little
Man,
frustrating
college
when that.
textbooks, you can just buy a new one for $700.
Oh my God.
I find it a little frustrating. I mean, like the taxonomy of a lot of groups is always changing, but it's frustrating when a term like zooxanthellae that has taken a while but has now become pretty recognized with the general public.
Right. Yeah.
And then for us to just be like, nope, we're gonna wipe that term off, and here's a new term.
Yep.
Why was it changed again? I'm sorry.
I honestly don't know. I don't know what the work behind changing the name was, but
I think Symbiodinium has been the genus name for a long time.
it
It's
has,
just,
yeah.
I don't know that zooxanthellae's not appropriate.
Our position had just went way off. Look at that.
Yeah.
Oh man. I wonder if we're at some climb or something.
Maybe.
Like Argus was great, and then just
just totally exploded up here.
Yeah.
Relax, guys. Argus didn't actually.
I know. I was like,
explode.
And what he means is everything is fine.
Just the USB out.
Twitter explodes too.
We can input a different sound speed if that might help. I don't know.
We can try that. I don't know.
While we're talking about kind of environmental impact, this question's come up a few times. Are we seeing
The EOS
any
is 1485.7.
like physical garbage or plastic bottles, any human kind of impact physically or only chemically at this point?
We haven't in these dives seen any like marine debris or things like that, and marine debris tends to get funneled
We're not that
around
worried
in
about
these
it.
like large ocean currents. So if you've heard of the Great Pacific Garbage
I know
Patch,
where
that's
I
a
am, and I know
big
where she is.
area of high,
And
a
we're
high
connected,
concentration
so-
of marine debris and marine plastics that
Hopefully
are
the
floating
meme
around
is
in these
still-
gyres that take place on the currents above and below the equator. So in the North Pacific Gyre is a big one that sort of swirls around a lot of marine debris that we see catching on the beaches, definitely in the Hawaiian Islands and the northwestern Hawaiian Islands. But I think we're a little bit below those currents.
Video shrimp.
So we're a bit out of those areas.
Shrimpy.
Oh, he knew.
And we do see garbage or trash on the seafloor as well. It's just like we haven't seen any on this cruise, which has been really nice. So indicative of our
Coming right at
targeted
us.
paths on ridges and
Alright.
steep terrain.
Yeah.
I love that.
But
Buddy,
But
calm
there's
down.
good evidence,
Just wanna
like
take
some
your picture.
recent studies are showing like microplastics pretty much
Outta
everywhere in the
here.
ocean,
Alright,
including in some of the deepest like trenches in the ocean.
yeah. And actually, there's a scientist, Randy Rogen, who was on the work we did in the Phoenix Islands, which is just south of here. And she took samples to look for microplastics in corals. She's still analyzing them, but it'll be interesting to see if she sees any evidence of that,
I
and
wouldn't
because
be
that
surprised.
is a pretty it's been a protected
deep sea
area.
corals?
Yeah. It's been a protected area
Video
for
push in.
a pretty long time.
Yeah, people are really surprised to find trash in the Marianas Trench, but it's actually, right adjacent to where people are living.
Right.
dumping,
It's also a
so-
trench. So
Yeah,
like
exactly.
Everything flows into there.
For some reason, it seems impossible for something to sink that deep, like trash, I guess.
Like a Oracle-less lobate ctenophore, like a Thalassocalyci.
Wow.
These are like really hard to sample.
Oh.
And so a lot of them are only known from image data. Because
Full
sampling
line.
these with a net, just you get debris.
They just
chunks, yeah.
disappear.
You touch it with your hand, it's like gonna.
Yeah, we tried to slurp some of them on my last cruise because we were doing midwater transects, and they came up just like the slurp was just a pile of jelly.
Yeah. When I used to do microscope work, there'd be like, you have all your vials written out, and there's one that's gelatinous debris, and that one tends to be really populated. And there's no ability to identify any of that stuff. That's why the imaging and to some extent the acoustics can really help narrow down some of the taxonomic, those spatial distributions for those kind of hard to sample taxa.
Yeah.
All right, we're near the part where we're going to start climbing again.
Cool.
Seems.
Yeah.
Nice.
How much time are we going to have to get up this slope here?
Half an hour.
Okay.
Do you want to look for a rock here?
Well, we kind of a little bit missed our shot. I feel like that we would have to go back to like there to get something good, or keep moving up.
So we're about 300 meters from the next summit.
We're not going to do that.
Yeah, we're not going to make it.
No. So
do we want to go back and get another rock from the other place?
I don't know. Alexis, what do you want to do?
No, yeah, I mean, I think the rock has priority now, so.
I guess I would say like maybe we could just do like a little like loop south and west of where we just came to kind of like circle around that, like that.
Just kind of follow the contour of the ridge. And then
Yeah, and like stay on top of the mound, but kind of on the other side of it.
Nice big
The
C
west,
pin.
Okay. I'm going to put in 220 and just 20
Sure.
minutes. Is that all right?
Yeah, you can call it in. I'm going to
And
get
you'll
lined
reposition.
up.
Okay.
It would have been cool to make it to the top, but if we won't make it, then we need to
it
get a
doesn't really
rock.
matter.
Yeah. And I think if we were expecting carbonate, we would have seen it already.
Yeah.
The caps are normally pretty thick, so.
I can go way faster in this direction than I can in the other direction.
Oh.
That's a plus. The USBL really did.
Watch as it comes back.
I know, that's what I'm curious about. I'm kind of interested in what our sensor gauges are.
There are all
over
these
this
C
transition.
pens.
Another C pin.
Oh, nice.
Yeah.
Does anyone know about the adhesive structure of the C pens that are sitting on the sediment?
No.
What do you mean?
Do we know?
Are they different from
Oh,
the
I don't know,
adhesive
I'm asking.
mechanisms,
Oh.
the ones that adhere to rocks or
Yeah.
something?
I don't know how different the peduncle actually is. I just know that it buries itself in the sand, and so it flattens out a bit underneath